Hi everyone, my name is Aurelia Reynolds and I am going to be presenting my senior thesis project, The Effects of the LON Protease on Topoisomerase 4 Activity and Caulobacter crescentus. So this is Caulobacter crescentus. This is what the cell normally looks like. And there's a few interesting things about Caulobacter that I definitely think is noteworthy going forward into my project. So one thing that you should know is Caulobacter crescentus are gram-negative bacteria with a very unusual cell cycle. During replication, cells go through a swarmer and stalk stages, where it starts off as a swarmer cell, goes through DNA replication, and ends up diverging into a stalk cell. This stalk cell can kind of be dormant and then end up actually going through cellular division. After cellular division, a cell can either go back into the stalk cell or can go into a swarmer cell. So you always have two sets of cells, one in the swarmer cell where it's actively going through DNA replication, and one in the stalk cell going through either division or kind of waiting in a almost dormant phase. The thing, now switching gears, I want to talk about the LON protease. LON protease is a protein that degrades other proteins in the cell. And through experiments, it has been able to deem certain genes as essential. Mutants that block a LON protease are called a delta LON, and I'll be referring to any mutants as delta LON for the remainder of my project. Now, going back to how these experiments are deemed certain genes essential. Basically, through experiments, we're able to knock out a LON protease. That means that the mutants delta LON do not have this LON protease. They do not produce it in any sense. And so there's a lot of different regulation effects that you can see. Through experiments called RNA sequencing and transposon sequencing, we're able to see some types of data. RNA sequencing is actually able to, which is what you're seeing on the top, right here and right here, you're actually able to level the measures of RNA expression through different genes. You're able to see it both and be able to compare it in a wild type and a delta LON. And then also you're able to have transposon sequencing. And transposons are introduced into the DNA, and they actually halt the DNA replication. They halt the RNA production as well. And so cells or genes with a lot of transposons, they're actually not able to be produced. And so if you're able to see a gene right here, for example, the wild type of this gene is actually considered non-essential just because there are so many transposons that are introduced into the gene. If a cell in the transposon sequencing data does not have any ticks like this, it means it's considered essential. That means that you are not able to introduce any transposons and have a viable cell. That means the gene has to be present in order for the gene to continue, in order for the cell to continue living. Delta LON mutants, we know that they have increased levels of DNA replication, they have altered cell cycles, and they have longer filamented cells. In this picture, you can see that the delta LON cells are on the right, and the regular Caulobacter cells, the wild type cells, are on the left. You can see how long and filamented they are and how they end up not becoming they're short types of bacteria cells. So switching gears, I want to talk about another protein called topoisomerase 4 and its role in DNA replication. All cells have topoisomerase 4 and is used for two types of activity in bacteria. One is to relax supercoils that are produced during DNA replication. Supercoils are basically tension that is created within a double helical structure when you open up the double helix for DNA replication, and your DNA replication apparatus moves throughout the double helical structure. The DNA actually ends up getting supercoiled. Think about an old telephone coil. If you twist that and twist that and twist that, it's going to bundle up in itself and produce coils on top of coils on top of coils. This creates a lot of tension on the DNA and causes sometimes it to break. So what topoisomerase does is it goes through, it clips either one or two of the strands, it untwists it, and it rebinds it so that the DNA, as the DNA replication apparatus is moving, the DNA can continue to be relaxed and not have that type of tension and stress. Another function of topoisomerase 4 in bacteria specifically is decatenation, which is what you can see on the bottom. after bacteria go through the entire DNA replication activity, they have these two interlocked chromosomes before they are able to completely divide. And so what topoisomerase 4 does is that it clips one of the chromosomes, it unlinks, it binds them together, binds them back to themselves, they unlink them, and then the two chromosomes are able to go into the cell division. Now, there are two genes that are helping with topoisomerase 4 regulation in Caulobacter. One is NSTA and one is GAPR. I want to talk about GAPR first. It helps with the topoisomerase 4 activity with the relaxation of supercoils. So what GAPR does is it recruits topoisomerase 4 to an area with supercoils, and together they help relax those supercoils and alleviate the tension. This is just a diagram showing the topo 4 decatenation activity again, because the next gene, NSTA, actually inhibits this. NSTA will go and bind to topoisomerase 4 and inhibit the decatenation activity of that protein. So after replication, it is not able to unlink those two chromosomes. And what's so important about this? Well, like I explained earlier, we were able to see through TN sequencing and RNA sequencing data essential and non-essential genes. This is what we're seeing for GAPR. So already we can see through the RNA sequencing data, through this green arrow, this higher level of RNA expression in a delta long mutant. And we're also seeing no transposons in the transposon sequencing data, which is inferring that NSTA is actually an essential gene in a delta long mutant. So in a delta long mutant, it is essential to the fact that NSTA inhibits topo 4 activity. Oppositely, in GAPR, which is what we're seeing right here, you're seeing, again, different levels of RNA expression between the wild type and delta long, but what's really interesting is through this black arrow right here, you're seeing the transposon sequencing data. Transposons are introduced, and therefore GAPR cannot be considered essential in a delta long mutant. However, it is considered essential in a wild type. Now, why is this interesting? We know that there's increased levels of DNA replication in delta long mutants. So why would the inhibition of topo 4, something that helps DNA replication, be inhibited, and the inhibition of topoisomerase 4 activity, something that helps in DNA replication, why would that be upregulated, but the activation of topoisomerase 4 activity would be downregulated? So what are some of the questions that we're trying to ask with this project? Well, first, I really want to know how topo 4 activity is regulated in Caulobacter crescentus. We know we have NSTA, we know that we have GAPR, but I just want to see how it fits in together as a whole. And then I want to look at how is that regulated in a delta long mutant. We already have seen that, NSTA and GAPR, those levels of different RNA expression, we're looking at the different types of whether or not they're essential. Want to look at how it's regulated differently in a delta long mutant. I'm also saying, is what we're seeing a direct result of lacking a long protease, or is it due to downstream effects? So is the upregulation of an NSTA protein, is that due to the fact that LON is no longer there to degrade it, and that would be a direct result, or is it due to a downstream effect? We know that LON has some, or a delta long mutant has some effects in the DNA replication with DNase, which is an entirely different protein, and is that what is causing this strange topoisomerase 4 activity? I want to see what other genes are important in the topoisomerase 4 regulation. We have these two already. There's the two subunits of topoisomerase 4. And so we're just trying to see what other genes are important in this type of regulation. And with that, are these genes upregulated and downregulated in the same fashion as NSTA and GAPR? So are we seeing inhibitory proteins upregulated and activating proteins downregulated? And all of this to ask our one big question: how does LON affect topoisomerase 4 activity in Caulobacter crescentus? So my hypothesis is that genes involved with topoisomerase 4 activity will be downregulated in a delta long mutant, while genes involved with topoisomerase 4 inhibition will be upregulated in a delta long mutant. And some of the phenotypes that we know are in delta long mutants, I believe can be correlated to this, like the longer filamented cells and the abnormal cell cycle. So why are we asking these questions? Well, there's really one big answer, and that is antibiotics. A lot of antibiotics actually use the inhibition of topoisomerase 4 to stop DNA replication in bacteria that cause disease. While Caulobacter crucentus does not cause any disease in humans, it's still really important to look at the topoisomerase 4 activity in a viable model. Why this is so prevalent is because antibiotic resistance is being seen more and more in everyday life, and many diseases caused by bacteria are becoming harder to treat. Hopefully, that by studying the topoisomerase 4 activity and helping to produce some information towards the study of antibiotics, we can help with antibiotic resistance. So how I'm going to go about this project is that I have a few couple methods. One, I have a statistical analysis of raw data. This is just basically an Excel sheet that kind of compiles all the genes in Caulobacter crucentus, this specific model, and kind of we talk about the p-value and whether or not we want to accept the differences in RNA or TN sequencing data. Obviously, we can sometimes see it with our eye, but sometimes our eye is not the best thing to rely on. So here we have the raw data that we're able to see if there is a statistical difference between the two or not. The second is through a viewer, which I showed earlier. We're able to see, the distinct differences of the TN sequencing data and the RNA sequencing data. And with this, you're able to see with your eye and then go back and confirm or deny with your statistical data. Also with that statistical data, we're able to go into RStudio and look at some plots. So you're able to see again whether those genes are upregulated and downregulated and whether or not you should accept that type of p-value. There's another type of network called the string network where you're able to see the connection of different genes and different proteins in this type of model and how they're connected. This is something what it looks like. You're able to click on each of those and kind of read about what the gene does. Par-C is actually one of the subunits of topoisomerase 4. It's responsible for the catalytic activity. So that means it's responsible for the decatenation as well as the relaxing of the supercoils. And you can see that it's connected to a lot of different things, like the Par-E, which is the other subunit of the topoisomerase 4, which is actually responsible for the enzyme part of it that creates the activity. It's also related to GIUB, DNAK, things that are known in DNA replication, as well as some other things. Lastly, literature review. There are a lot of papers out there about Caulobacter just because it's a great model to be using. However, there are not a lot of papers out there about NSTA or GAP-R for that matter. But it's still there's a lot of information out there, and it's great just to analyze it as we can. My results are to be determined. Still working on through my hypothesis, so they can't be explained really right now. And I really want to have a discussion based off of these results. I want to be able to say, what do my results infer? I want to go over, possible areas of future direction, possible questions that are coming up from these results, and really be able to walk through the process of this experiment. I do want to have the time to explain that I will be available for questions through my email, or maybe hold a Zoom question hour time. I'm not really sure how the conference is going to be going, but I do want to have a time for questions and acknowledgements. So thank you, everyone. This was great. Thank you.